Hello everyone, my name is Gary O'Mealy, and in this video we are going to discuss pH and buffer systems in human physiology. Every solution that uses water as its solvent is going to have a measurable pH value, and since in a previous video we discussed how every kind of body fluid that you find in the human body uses water as its solvent, this is going to be applicable to just about everything that we talk about throughout this course. The reason why every kind of aqueous solution is going to have a pH value is because of the natural but not too common tendency of water to ionize in solution. Here you can see a container of water in which water ionizes into a hydrogen ion and a hydroxide ion. So in a container of pure water with no solutes, no additives, no nothing, the concentration of each of these types of ions is going to be 1 × 10 to the -7th moles per liter. So very, very, very low concentrations for both hydrogen ions and hydroxide ions in a container of just pure water. But the idea here is that adding any kind of solute to an aqueous solvent can cause these concentrations to change over time. And since we've discussed in previous videos how the different bodily fluids contain solutes like sodium and potassium and things of that nature, we need to be aware of how adding these sorts of solutes into a solution can cause the pH to change. So we define any solute that causes the hydrogen ion concentration to go up and the hydroxide ion concentration to go down, we call those acids. And then consequently, any solute that causes the opposite reaction to happen, being that hydrogen ions go down and hydroxide ions go up, these are called bases. So it's the concentration of these ions that are going to end up being very important for a lot of different chemical reactions that happen inside of living systems. And obviously we have, in a previous video, discussed all sorts of metabolic reactions. So we can actually quickly quantify the concentrations of both hydrogen and hydroxide with a pH measurement. The most common formula that you're going to see associated with pH is this one right here. The pH of any given solution is equal to the negative log base 10 of the hydrogen ion concentration. We can also calculate what is called the pOH, which is almost exactly the same sort of formula, negative log base 10 of the hydroxide ion concentration. And in both cases, the brackets here indicate that we are talking about the molar concentration, that being in moles per liter. Now, the reason why I have the pH formula here highlighted in green is because this is going to be the one you use in 99.9% of situations. There's usually not going to be much of a need for you to calculate the pOH of any given solution. But we can see that when you add the pH and the pOH of the same solution together, you should always get the same number, that being 14. And mathematically speaking, this should give you an idea of the relationship between hydrogen ion concentration and hydroxide ion concentration. When one goes up, the other goes down. So practically speaking, the pH scale is going to run from 0 to 14. In theoretical terms, it's possible to have a pH lower than 0 or higher than 14, but in living cellular systems, we're not going to need to worry too much about that. So the idea of how this works is that solutions with very low pH values are actually going to have very high hydrogen ion concentrations, and we consider those types of solutions to be very acidic. Similarly, solutions with very high pH values will have low hydrogen ion concentrations, and we'll consider those solutions to be very basic. On the far right here, you can see a figure from the textbook showing you how different types of everyday substances have a variety of different pH values associated with them. Battery acid obviously is very acidic. Citric juices, lemon juices, and things of that nature will also be very acidic. Pure water is going to have a neutral pH of about 7, and then as you go up towards the basic side, you can see some examples of some more alkaline substances that you might find around your house. Things like ammonia, baking soda, oven cleaner, and things of that nature. The main thing I want you to understand about the pH scale itself is that it is logarithmic. So if you're comparing two pH values, being aware of the logarithmic nature of this scale is going to be very important for understanding the magnitude of how pH changes, especially when those changes don't seem to be very big. So because this is a logarithmic scale, a difference of 1 pH unit actually means a 10-fold difference in hydrogen ion concentration. For example, if you were to compare the pH of pure water, which is 7, to the pH of seawater, which according to this figure here is 8, that would indicate that pure water is 10 times more acidic than seawater because they have a difference of 1 pH unit. Or you could interpret this the other way and say that seawater is 10 times more basic than pure water. Similarly, a difference of 2 pH units will mean a 100-fold difference in hydrogen ion concentration. So this time, if we are comparing coffee, which has a pH of 5, to pure water, which again has a pH of 7, we could say that coffee has a pH that is 100 times more acidic than pure water. So that is the nature of this logarithmic scale. The more pH units separating two different solutions, the bigger the magnitude is in terms of differences in hydrogen ion concentration. So, like I said, understanding how this scale works is really going to be important because in the human body, you're not going to see pH changes that happen in whole units of 1 or even 2 units. You're going to see what appears to be very small changes in pH values, but I am here to convince you that a very small change in the pH can still mean very big and very significant changes in hydrogen ion concentration. So let's look at an example of this. The blood plasma has a set point pH of 7.40, and that's a number you might want to go ahead and commit to memory because that's going to be a very important number as we go throughout the course. So we're going to ask the question, what if the pH of the blood is allowed to drop to 7.20? Now, a difference of 0.2 pH units may not seem very big to you, but we're going to use a little bit of quick math here to show you that it actually is a very big difference. So at a pH of 7.4, we can do a little bit of quick algebra to show that the hydrogen ion concentration of blood plasma at a pH of 7.40 is 4 times 10 to the minus 8th molar. Now, if we allow the blood plasma to dip all the way down to a pH of 7.2, then doing the same sort of algebra here would show us that our new hydrogen ion concentration would be 6.3 times 10 to the negative 8th molar. Now, if we take the ratio of these two numbers, 6.3 times 10 to the minus 8th, which is the hydrogen ion concentration at a pH of 7.2, and put it over 4 times 10 to the minus 8th, which is the hydrogen ion concentration at a pH of 7.4, we get a number of 1.575. So basically, our interpretation of this is that at a pH of 7.2, the blood plasma would contain 57.5% more hydrogen ions than blood at a pH of 7.4. Now, you don't need a math degree to know that when something increases by 57%, that's actually highly significant, and that's actually a very, very, very big difference. And later in the semester, we're going to learn how changing pH actually affects all sorts of physiological processes. So you can hopefully at this point, even this early in the course, you can hopefully understand how it's very important that we do not allow the pH of body fluids like the blood plasma to change very much at all. Now, for now, we can understand that we don't want the pH of any body fluid to change too much because the pH will affect the sorts of things like the ability of hemoglobin to carry oxygen, which is called the Bohr Effect, the stability of proteins, which is something that we'll illustrate in a later video, and 3, the efficiency of enzyme-catalyzed reactions, which again we will look at in a later video. So if the pH of the blood plasma were allowed to dip from 7.4 to 7.2, all 3 of these things might change for the worse and body function would be negatively impacted. Now, just to give you a quick illustration of how the Bohr effect works, what you're looking at here is the oxygen saturation curve for hemoglobin at a pH of 7.4. The y-axis here is showing you the percentage of all available hemoglobin molecules that are fully loaded up with oxygen. So at the very top of the graph, you would have all or 100% of your hemoglobins loaded up with oxygen, and then at the very bottom it would be 0%. So on the x-axis, we are looking at the oxygen saturation in the blood plasma, and the idea here is that at very high oxygen levels, hemoglobin gets close to 100% saturated, and at low oxygen levels, hemoglobin is very fractionally saturated. Now, the reason why I have a graph here dedicated to a pH of 7.4 is because this is how hemoglobin behaves at that particular pH value. But as we said, hemoglobin will behave very differently when the pH changes. If you change the pH down to 7.2, the graph shifts to the right. If you increase the pH up to 7.6, the graph shifts to the left. So our interpretation here is that a leftward shift means that hemoglobin will now have a much higher affinity for oxygen. It will bind much more tightly to oxygen, and it won't want to let go as much. A rightward shift in the graph similarly means a lower affinity for oxygen. Hemoglobin will have a much harder time hanging on to it. Now, the green graph at a basic pH might seem like a good deal, but it's important to remember that hemoglobin not only needs to be able to bind oxygen and carry it around, but it also needs to be able to let go of oxygen and deliver it to respiring cells when necessary. So what this should illustrate for you is that hemoglobin's oxygen binding dynamics are actually perfect at a pH of 7.4, and allowing the pH of the blood plasma to change either down to 7.2 or up to 7.6 would produce undesirable effects on the ability for hemoglobin to carry oxygen around. So the question becomes, well, how does the pH of something like the blood plasma change over time? And the thing you need to remember is that there are thousands of different metabolic reactions happening inside the body at any given time. And these reactions will constantly be producing chemicals that can potentially shift the pH at any given moment. Remember that when you add a chemical to a solution, it can change the pH. Whether it acts as an acid or a base is the question. So the best examples to illustrate how metabolic reactions can change the pH are those reactions in cellular respiration, and those reactions are going to produce a lot of carbon dioxide. So if we look at the overall reaction scheme for cellular respiration, we can see that glucose reacting with oxygen gives off carbon dioxide and water. So we know for sure that neither oxygen nor carbon dioxide are very soluble in water. That's something that we talked about in a previous video. Oxygen gets around this solubility issue by being carried around by hemoglobin, but the question becomes, if carbon dioxide is not very soluble in water, then how do we carry it around? How is it transported throughout the body? Well, one of the ways we get around this is by using an enzyme-catalyzed reaction that will actually solubilize carbon dioxide. This enzyme is found in red blood cells, and it is called carbonic anhydrase. You can see the reaction that is catalyzed here takes carbon dioxide and water, the two major byproducts of cellular respiration, and converts it into carbonic acid, H2CO3, which, as a weak acid, will partially ionize into a hydrogen ion and a bicarbonate ion. In either case, bicarbonate ions and the fully fledged carbonic acid are both very soluble in solution, so that helps to solve the issue of how are we going to transport carbon dioxide around. We are going to temporarily turn it into something that is very soluble. But the issue here is that in doing this, we are releasing free hydrogen ions into solution, and that's going to significantly lower the pH in the vicinity of cells that are performing cellular respiration. So the Bronsted-Lowry definition of an acid, you may remember this from general chemistry, is any chemical that releases or donates hydrogen ions or protons into solution. So that is how we would nail that carbonic acid is indeed an acid, other than the fact that it is literally called an acid. So if we look at a quick little illustration of how this all works, you can see here in the blood plasma, a red blood cell is carrying oxygen around. This oxygen is delivered to a cell that is performing cellular respiration. And in a later video, we'll detail all the different chemical reactions that happen during cellular respiration. But in particular, the one that we're interested in are all the reactions of what are called the Krebs cycle, which happen inside of mitochondria. These Krebs cycle reactions give off a lot of carbon dioxide. This carbon dioxide will leave the cell and get into the blood plasma, and this carbon dioxide will diffuse into the red blood cells, which contain this carbonic anhydrase enzyme. And then as carbonic anhydrase solubilizes the carbon dioxide by producing carbonic acid and bicarbonate, we produce these hydrogen ions, which, as we said, produce the undesirable effect of lowering the pH of the solution. So the idea here is that the more carbon dioxide that gets produced, the more hydrogen ion gets produced, and the lower and lower and lower the pH is going to become. So blood plasma, we get around this by using what we call buffering systems. So the blood plasma is buffered by salts, proteins, and then also the removal of excess carbon dioxide by using the respiratory system. Once that carbon dioxide is solubilized in solution, it travels through the bloodstream, is eventually reconverted back into carbon dioxide in the lungs, and then we're able to safely breathe it out. So the body actually maintains pH homeostasis in a variety of different ways. We just mentioned, number one, the expulsion of excess carbon dioxide by the respiratory system, and that is something we will talk about in much detail later in the semester. Any excess hydrogen or bicarbonate ions can be secreted or reabsorbed to or from the blood by using the kidneys. And then, of course, we use the buffering systems that I mentioned before, salts and proteins that are found inside of the blood plasma and other bodily fluids can help to resist changes in pH caused by all the different metabolic reactions that we've discussed. So that's going to pretty much do it for this video. Here is a list of vocabulary terms you probably ought to be familiar with after watching this video. And then for checking your understanding, number one, what is pH a direct measurement of? Number two, why are small differences in pH actually very highly significant in the grand scheme of things? Number three, how does carbon dioxide affect the pH of bodily fluids? And number four, how is pH homeostasis maintained in the body? So that's going to do it for this video. Thank you for watching, and if you have any questions, go ahead and leave it in the comments section. I will see you next time. So long.